They will also eat anything else that happens to fall in the way that's proteinaceous, and I'm pretty sure they would eat you if you fell in the way as well and were unable to move. Now that gave us a really interesting insight. What we have here is a nutrient, a specific nutrient, an important nutrient, protein, whose deficiency has this massive effect at the population level, which stems back to individual protein deficiency in crickets. And we were able in the field to do experiments where you manipulated the amount of protein animals were given over a short period, and then you fed them a victim and saw how much they ate. And it was real schoolboy stuff. And we were able, therefore, to demonstrate that this specific appetite for protein was having this massive collective effect at the individual level. And that took us back to our earlier work with locusts, where we had also shown in those that they too have a specific appetite for protein. And in fact, we went on to show that locust swarms too are driven by cannibalism. And I thought this guy was particularly prescient in this B-grade book written in the 1950s. In the case of the Mormon crickets, there's a really interesting problem that the Utah and Idaho state traffic authorities face. When they start crossing the road, they get run over and they get eaten by other crickets who get that run over, and then before you know it, you've got this sort of ankle-deep slurry of grease across the road, and they have to send out the snowplows to clear them. Anyway, we've now segued into protein as a key nutrient. And as I said, if you take a locust has an extraordinary, we demonstrated this in experiments, an extraordinary capacity to regulate its intake of protein very precisely to a target point over a given period of time, and it will do so independently of other nutrients, particularly carbohydrate, which is their major other energy source in their food. And if you challenge them by diluting their food or by giving them different pairings of foods, if they can, they'll achieve that target point. And we did experiments to show how they do that, the sorts of amino acid feedbacks in the blood that are involved, the way their taste responses change, and so on and so forth. But that's not the key point that I want to make here. The point I want to make really is what happens if the animal's not able to get to its protein target. So if you allow it to, it will go there. But if it can't get there, what happens? Well, cannibalism is what happens in one instance, and that can lead to mass migration and swarming and locust outbreaks. So that's one problem. But what happens at the level of the individual? Well, there's an optimal diet for a locust, and if you give it that, it'll get to its target point, which I've shown here over a couple of days in its life, and it'll stop. It doesn't eat any more, it doesn't eat any less, it gets to there. If you confine it to foods that contain a lower proportion of protein, and these are the steeper lines here, or a higher proportion of protein, what you'll see is that on low percent protein diets, they'll overconsume carbohydrate, this dimension, to a greater degree than they're willing to overconsume protein on a high percent protein diet. The shape of that relationship, or that array of points of intake, is a fundamental referent in the study of nutrition. And that's one of our major contributions to understanding nutrition in general, is that we need, as biologists and human biomedical scientists, to consider the shape of this as well as the position of this. That's the requirements of the animal. This is how it compromises when it can't get to there when faced with imbalanced diets for whatever reason. Well, keep that signature shape in mind, elongated in this direction and shorter in this direction. So you've got a smear in the vertical direction. Carbohydrate varies more across those diets than does protein. Now, there are associated costs. If the animal is on a low percent protein diet, we found that they became obese and died early. So the take-home message from the shape of that array is that protein intake is prioritised if the diet forces the animal to have to trade off protein and carbohydrate intake. We found the same pattern in rodents. Just check out the shape of that. That's their intake target. If they're allowed to get there, they will. If they're not allowed because they're confined to imbalanced diets, you get a very similar sort of a shape. It develops over time. We found it also in mice, in fish, and in birds. So it seems to be, particularly among omnivores and herbivores, it's a signature pattern that you find in the nutritional biology of many animals. We also found it in humans. Now, the first experiment we did with humans, we published some little while ago now, in 2003, and it involved, this is when I was still at Oxford, we took a bunch of undergraduates up to the Swiss Alps. We incarcerated them in one of the undergraduates' parents' chalets up in the Swiss Alps, and we gave them buffets of food items, and they were allowed to eat whatever they liked from these buffets for breakfast, lunch, morning tea, afternoon tea, and dinner. And we measured how much they ate, and we confined them for 2-day periods to high or low percent protein items, or a full choice of all items on the buffet. And what we found was that when they were on the lower percent, when they were free to choose, that's where they ended up in a protein, carbohydrate plus fat space. Now I'm combining fat and carbs here. Fat's not really an issue for locusts, but it is for us. I'm just going to combine them as a single non-protein energy axis, because you get most of the signature you need out of it, actually, but that's another story. So, if allowed a free choice, our subjects went here. When they were confined to low percent protein buffet items for a couple of days, they overate relative to that, particularly in this dimension, hardly at all in the protein dimension. When on a high percent protein regime, they underate substantially in this dimension and hardly overate protein at all. You're starting to see that signature shape again. We then went back to the literature and plotted other studies which we could reinterpret that lasted for anything up to 6 months or a year, and just put their data on top of ours, and you see the same pattern emerging. Alison Gosby in the lab has just done a larger comparative analysis or meta-analysis which comprises of 30 or 40 studies, and this pattern is becoming stronger and stronger and stronger. If we plot those same points in a slightly different way, you'll see the following pattern. So what I've got here is the percentage of protein in the daily diet. 10%, 20, 30, 40, 50 is a percent of total energy. And this is the absolute amount of nutrients eaten, of protein and non-protein energy. And what you'll see is as the percent protein in the daily diet increases, there's hardly any change in the amount of protein eaten. It's remarkably flat. But the amount of non-protein energy shoots up as you decrease protein intake. That's a really strong pattern. But we don't know whether that's just some peculiarity of people knowing what they're eating, the foods being familiar, the circumstances of the experiments and so forth. And to get a really acid test of what we're calling the protein leverage hypothesis, we needed to disguise the macronutritional quality of foods, to confine people for week-long periods on diets where they thought they were eating the same variety or a wide variety of different foods, which were in fact all exactly the same. Palatability matched, so they just had the feeling of eating ad libitum in abundance, and for the different weeks of the study, they had different percentages of protein, 10, 15 or 25%. The subject couldn't tell. Alison ran this study here in Sydney. We're currently running another in Jamaica. We've run one in the Gambia, and there's another one recently finished in Cambridge, which we've all coordinated from here. Alison's data are, I think, really quite remarkable. So just to summarise before I show you those, it seems too that we humans prioritise protein when our diet forces a trade-off. Is it the case that we do this without any knowledge of the nutritional quality of our food? Well, here's a 10% lunch and a 25% lunch from Alison's study. You'll see they're indistinguishable by eye, and they're actually indistinguishable in terms of their taste and palatability as well. If we superimpose the data that came from that study, which had just in the last couple of weeks been analysed fully, there they are. So that's proof that the protein leverage effect is real. We humans prioritise protein intake when our diet shifts, for whatever reason, the proportion of protein to non-protein energy. And it's not just we human primates. We had the good fortune to work with a PhD student from ANU, Annika Felton, Swedish girl who'd done this brilliant study, field study in Bolivia, where she watched through binoculars individual spider monkeys for full daylight periods. And each of these points is a full day's observation for a single monkey. And what she did was she recorded everything a monkey in the tree put in its mouth, which bits it spat out, chemically analysed all the different sorts of foods, and from that we were able to construct a protein, non-protein energy plot of the same sort that I've shown you for humans already. So there's the human one, percent protein in the daily diet, nutrients eaten. These animals maintained their protein intake absolutely constant from day to day. Their non-protein energy intake varied over 7-fold as a result of the fruits, of the leaves that were flushing in the cafeteria of their environment from day to day. There's at least part of our ancestry for our protein leverage effect. What are the implications? Well, if the diet shifts to contain a higher proportion of carbohydrate and fat, and particularly if it's energy dense, then the protein leverage effect is going to cause us to overconsume non-protein energy in an effort to gain limiting protein. We have no conception that this is going on. Our bodies are doing it below our level of consciousness. Now that poses us with a problem, an energy imbalance. Unless we get rid of that excess, it's going to change our body composition. And just to give a feel for the power of that effect, if you go back to, say, the US population in 1960, estimates were that they had a 14% protein by energy diet on average. By the year 2000, that had dropped 1.5% to 12.5%, which sounds pretty trivial. And that was largely the result of eating more carbohydrate and fat. However, they maintained over that period their protein intake constant. And that's been exacerbated quite clearly by a reduction in energy expenditure associated with driving cars, watching television screens, having centrally heated houses and so forth. In contrast, if you shift the diet in this direction, then you're going to end up under-eating energy and potentially therefore losing weight, which explains why all the high-protein diets work, to some degree at least. But it may well pose costs. And these in the human population are yet not clear. But it is a really interesting question that I'm going to come back to in just a couple of minutes. There's also, it would seem, a catastrophic positive feedback. Now let's say the diet shifts towards incorporation of more carbohydrate and fat. Now there are many asymmetries in our modern world which encourage that. Partly economic, it's cheaper to buy carbohydrate and fat-rich foods. Linked to that, it's associated with an increase in processed foods in the diet, which are also by definition higher in carbohydrate and fat. We also have an ancestral predisposition to find fat and carbohydrate really attractive, and that probably stems back to our Paleolithic ancestry where those nutrients were rare. But there are many reasons why, despite living in a world of abundance, we're tempted to go in this direction and hence overeat. Once that happens, there's a predisposition towards obesity and insulin resistance. That's very well understood. Under those circumstances, we find that fat stores, particularly deep fat stores and intramuscular stores, start to burn themselves at a higher rate than they should, given that we're clearly in a positive energy balance. Free fatty acid levels, for example, in the blood rise, and that's a well-known associated condition with metabolic disorder. Those free fatty acids interfere with the inhibition by insulin of protein breakdown in muscles. Normally you don't burn your protein in your muscles if you're well fed. That's a last resort starvation response. Obesity and insulin resistance starts to encourage that. It also encourages the clearance of those liberated amino acids by subjecting them to a process called gluconeogenesis in the liver. The long and short of it is that you start to burn protein, and that is going to increase your protein requirements, because your hunger system for protein, powerful as it is going to be saying you need more protein. So it's going to shift your target this way, and that's going to cause a terrible positive feedback, because if you stay on the same diet to get that extra protein, you've got to eat even more. So you kick in to a vicious metabolic cycle. Well, let's just come back to that question. Why don't we overeat protein? Why do we have this tendency to not allow ourselves to undereat it? That's clear. It's pretty clear why you shouldn't eat too little protein, because it's essential for growth and for the replenishment of broken down proteins and enzymes. Because protein contains nitrogen, and nitrogen is an essential element for all of those processes. So you can understand perhaps why animals don't like eating too little protein and might up their intake to gain it. But why don't you overeat it, particularly if you're an omnivore or a herbivore? There's another issue there with carnivores you might have thought about. I'll talk to any of you about that later if you like. We've addressed that problem recently. Sean Wilder in the lab has just got a very nice paper coming out on that. Well, if again taking an evolutionary biology perspective, you might think if animals are designed not to do it, there may be some cost to doing it, over-consuming protein. In which case, that may ring alarm bells in terms of human diet. Well, to try and understand what the costs of eating too little or too much protein or too little or too much carbohydrate are, we decided to turn to our friend the fruit fly. Beloved of geneticists around the world, this little tiny animal has been one of the most significant creatures in the study of biological science. What we did here was we confined over 1,000 fruit flies to one of 28 different diets, varying in their protein to carbohydrate ratio and concentration. And we measured how much they ate, or indeed drank, because that's how a fly imbibes its food, by providing them in 5 microliter, very tiny pipettes, and measuring how much the meniscus went down as the animal drank. And that way you could quantify intake. So Fiona Clissold in the lab and Naz Saran and Kwang Lee, who's currently in Korea, spent many, many happy hours doing exactly this. Now, what that allowed us to do was, because we knew how much each fly had eaten, how much protein, how much carbohydrate, we knew how long it lived and how many eggs it produced, we could relate nutritional state to performance, particularly lifespan and reproductive output. And you can plot those as surfaces. And here are 2 surfaces from that experiment. This is protein eaten, carbohydrate eaten, and this surface here is constructed based on the individual flies that contributed to the experiment. And that landscape is a colour-coded landscape, a little bit like isoclines on a topographic map. The higher the surface, the redder it is. So what we have here is the summit in this region falling away, the slope falls away here to the deep blue and the lower elevations. Here's the equivalent surface for the number of eggs produced over the animal's lifetime. Now, what you can see from this is that protein is needed for growth and reproduction, but too much shortens lifespan and indeed shortens their reproductive output or reduces their reproductive output. So if you look at this surface here, as you move from left to right, you're increasing the proportion of protein in the diet, and the flies are living progressively less long. To maintain optimal or maximal reproductive output, they needed a higher intake of protein, but if you exceeded that, they started to lay fewer and fewer eggs as well across their lifetime, partly because they were living less long. If you gave flies an opportunity to mix their own diet, where do they go? Do they want to go to this point here and live the longest that they can, the sort of Methuselah fly, if you like, or do they want to maximise their reproductive output? Like nutrient-seeking missiles and good Darwinian animals, they went to maximise reproductive output. Now, there's actually a really interesting a side effect from this experiment. I don't know if you've ever heard about the caloric restriction lifestyle, which is a particularly miserable lifestyle, where people reduce their calorie intake by 40% in an effort to live longer. Well, it's predicated on this dogma, probably the central dogma in gerontology, that calories, not the nutrients from which they come, are responsible for establishing lifespan. If you eat too few, you obviously die soon. If you eat the right sort of amount, you'll live longest. If you eat too many, you'll start to live less long again. Now, bear in mind that protein and carbohydrate are what's called isocaloric. They have the same caloric density, which means that anywhere along a diagonal line is the same number of calories eaten. So that's a higher intake of calories, that's a lower intake, but it doesn't matter whether they're made up of protein or carbohydrate, they're all calories. That's the caloric restriction prediction, that's the reality. In flies, at least, caloric restriction does not prolong life. It's the balance of protein to carbohydrate that matters. Now, that's bad news if you happen to be one of these people who's currently making themselves very miserable by eating substantially less than they ought to. The same pattern has emerged from multiple other studies we've done with other insect systems, and the acid test is to do the same experiment with rodents. And we have now what's the world's largest nutritional study ever undertaken, macronutritional study, underway at the Anzac Institute here. Colleague David Lakuta, Sam Solon and others who are working with us on that project. We have here 900 mice housed on one of 30 different diets, which have protein, carbohydrate and also fat varied. So fat is an important dimension to them as it isn't for flies and locusts. And we're aiming, and the first cohort of mice will be sampled in a couple of weeks' time, to plot the same sort of landscapes for obviously things like how long they live, but also the associated changes occurring in metabolic and molecular pathways, which will underpin that phenotypic outcome. We're looking at the phenotype again in a really sophisticated way in an attempt to associate phenotypic outcomes, in this case nutritional outcomes, with the underlying molecular and genetic mechanisms. So where are we in the final frontier? We've started to work a little bit on genes, we've looked at developmental effects on phenotypes, we've looked intensively at describing in a sophisticated way the phenotype, and we've shown that phenotypic effects can manifest themselves in extraordinary ways at the level of groups and populations. We haven't yet, however, got to communities and ecosystems or the, as I said, abundant feedbacks. Can we get at those by concentrating on the phenotype? Well, that's where we're aiming next. And the way we're going to do that is to take some of the clues we've gained from the experiment so far and some of the techniques we've used to start to simulate not individual particles and locust swarms, but entire communities of individual species interacting. And the way we can do that, and we're currently in the process of commencing this work, is to use these individual-based self-propelled particles models in conjunction with our nutritional models and to put them into simulated environments where we can change the distribution, the abundance, the gradients of different resources and the numbers and composition of species within the habitat. That, I hope, will be the subject for a talk in a couple of years' time. At the very least, I think. All right, so what I've done then is I've taken you from paintbrushes to cannibals to obesity to aging, and I have too many people to thank that I could fit on the slide. There's a particular thank you to some of the key colleagues whose work I've mentioned here already, and our funding agencies. There are many, many others, students and postdocs over the years, both in Oxford and latterly in Sydney. It's been a wonderful journey, and it promises to take us in even more exciting directions. So thank you very much for listening.